is how. See, we know that we have purines and pyrimidines. So the degradation of either purines or pyrimidines will give out urea and uric acid. For example, these purines will give uric acid, whereas these pyrimidines will give urea. Now, the purine nucleotides are degraded by a pathway in which they lose their phosphate through a reaction of nucleotidase. So nucleotidase is an enzyme which can cleave the phosphate, because a nucleotide has a phosphate moiety, a sugar moiety, and the nitrogen base. So the combination of all these three things will only be called as a nucleotide. If there is no phosphate, then we call it as a nucleoside. If there is no sugar moiety and phosphate moiety, we call it as nucleic acid base or nitrogen base or purine base or pyrimidine base. That's all. We don't call it of any other names. So only when we call it as a nucleic acid, nucleic acid is a polymer. Nucleotide is a monomer. This nucleotide is having three subunits: one phosphate moiety, one sugar moiety, and a nitrogen base. If there is no phosphate moiety, we call it as a nucleoside, because it is a combination of a sugar as well as a nitrogen base. So this nucleotidase is an enzyme which can cleave the phosphate moiety. Now what happens is AMP or adenylate will yield adenosine. Adenosine means sugar plus nitrogen base, adenine. So which is deaminated to inosine. Sorry. So we know that from inosine we are going to get adenosine. This is done by this enzyme. So this is a deamination reaction. So this is adenosine deaminase, and this compound inosine is hydrolyzed to hypoxanthine. So this is done. This hypoxanthine is a purine base and D-ribose. So this IMP is broken down into two parts. One is hypoxanthine as well as ribose. So these two combine to form this ionosine. Now hypoxanthine is again oxidized successively to xanthine and then to uric acid. So this is a xanthine, what we are seeing in case of protein testing, xanthoprotic test. There is xanthine, and then ultimately we are going to get this uric acid. So this is done by the enzyme called as xanthine oxidase. So then there is a flavoenzyme which is in the form of this molybdenum, and there are 4 iron sulfur centers in the prosthetic group. So this enzyme is having this kind of features. So they are having 4 iron sulfur centers, as well as it is a flavoprotein. It's a flavoprotein or flavoenzyme, and this is having the central atom as molybdenum. Now the molecular oxygen is an electron acceptor in this complex reaction. So this is an oxidase. Here molecular oxygen will act as a electron acceptor instead of NADP or NADPH. Now this GMP catabolism also yields uric acid as an end product. So in the previous slide we have seen about this adenosine. Now we are seeing about this guanosine. So GMP is first hydrolyzed to xanthosine, so which is ribose plus guanine. Now this is then cleaved to free guanine. So here the ribose is cleaved off. This guanine undergoes a hydrolytic removal of its amino group to yield xanthine. Again this will give xanthine. With the help of xanthine oxidase, this again is converted to uric acid. So we know that uric acid is an end product of purine metabolism or catabolism, whereas urea is an end product of pyrimidine nucleotides breakdown. Now, so the catabolism of purine nucleotides. So we have two purines. One is the GMP as well as the AMP. So this GMP, so first of all there is a nucleotidase, so which will break the phosphate moiety. So if GMP there is no phosphate, then it is guanosine. This guanosine is again acted upon by an enzyme which is called as nucleosidase. See, nucleotidase, because it is a nucleotide, nucleosidase. That's the difference between the nucleotidase as well as the nucleosidase. So this is a nucleotide. This is a nucleotide which is having ribose, guanine as well as phosphate. First of all, this phosphate is being ripped off. So it is being cut off. In second case, ribose is being cut off. Now there is a free guanine. There is a free guanine. Now if we talk about this AMP, here also the same thing. First of all, there is a nucleotidase. You can see like this. So this hypoxanthine, which is from the previous slide, as well as we have a guanine from the previous slide. Now these two are converted into xanthine. This is a keto form. This xanthine, with the help of an enzyme called as xanthine oxidase, will convert this hypoxanthine or xanthine into uric acid. So uric acid is the ultimate product of purine synthesis, because you can see there is a purine nucleus. There is a purine nucleus with 4 nitrogens and all. There is no much difference between the xanthine as well as uric acid. You can see where is the difference here. Yeah, here there is an OH group. Here there is no OH group. Now this xanthine is converted into uric acid. If this uric acid accumulates, then we get a condition called as gout. Now what happens? Our body has a mechanism to convert this uric acid into urea. So we'll see that in the next. See, this is the uric acid. This uric acid is excreted as such in case of primates, birds, reptiles and insects. Now this uric acid is converted into ammonia as well as urea. So you'll see how it is converted. Now what happens is, there is an enzyme called as urate oxidase. This urate oxidase is converting uric acid into allantoin. So uric acid is converted into allantoin by the help of an enzyme called as urate oxidase. Now this allantoin is further converted into allantoate. It's converted into allantoate by the action of this enzyme called as allantoinase. So allantoin is broken down into, see here in the uric acid, what's happening here, this bond is broken first. So it's a cyclic form, and this is a binuclear cyclic form, and this is a mononuclear cyclic form. Now first of all, the bond between the CO is broken down, CO and NH2. So this is broken down. In the second case, even this CN is broken down. Now this ring structure, which is having 2 nucleus, is becoming 1 nucleus, and then it is becoming an open structure. So uric acid is a cyclic form which is having 2 nucleus, and allantoin is a cyclic form which is having only 1 nucleus, whereas allantonate is a compound which is having open structure. So first of all, the bond between CO and NH is broken down. In the second case, CO and NH is again broken down. Now ultimately we are going to have, this is CONH2 and this is CONH2. So we have 2 urea here. Now what happens? This allantonate is again converted into urea by the action of an enzyme called as allantolase. Now this part and this part is broken down into 2 urea, and what is remaining here? This part, the CHCOO, is remaining here, acetate. Now what happens? This acetate is removed as glyoxate. This is removed as glyoxate by action of water. So you can see here COOCH. So COOCH. So since it is water, it is converted into H plus and OH minus. So we require an oxygen. This H is removed and this OH is attached here. So ultimately we are going to get 2 urea and 1 glyoxylate. So this glyoxylate is left over, and this urea. Now this urea is further converted into ammonia by the action of urease. Now I have shown different different forms of the excreta of different organisms. For example, some of these birds or reptiles or insects or some of the primates, they can directly eliminate or excrete uric acid, whereas most mammals will excrete allantoin, and some of the bony fish, they will excrete allantonate, and so on. So human beings, we excrete in the form of urea or uric acid. Next, moving further, these pathways for the degradation of primates generally leads to ammonium ions production and thus the urea synthesis. So we know from ammonia we can synthesize urea, so by the urea cycle. Now this thiamine, for example, is degraded to methyl malonyl semialdehyde, so which is an intermediate of the valine catabolism. Even valine also has this intermediate. Now it is further degraded through propenyl CoA and methyl malonyl CoA to succinyl CoA. This succinyl CoA can enter into the Krebs cycle. So we know that one product can be catabolized and the intermediates can be shunted to the remaining metabolisms, like that. So through valent catabolism we are going to get this methyl malonyl semialdehyde. This methyl malonyl is again converted to succinyl CoA, and this succinyl CoA can be shunted to this Krebs cycle. So we know that this succinyl CoA can be shunted through the Krebs cycle, and this can be done by this metabolism. Here is the catabolism of pyrimidines, and thiamine, especially thiamine, I have taken the example of thiamine. So this is thiamine, and thiamine can be converted into dihydrothiamine. This can be done by dehydrogenase enzyme. This is called as dihydrouracil dehydrogenase. So this can, you know that thiamine and uracil can be shifted. I mean, they both are the same. Just a substituent of thiamine is a uracil. So, sorry. So this thiamine can be converted into dihydrothiamine, and this dihydrothiamine can be converted into beta uridoisobutyrate. So this is an open structure. So you can see here, this is a cyclic structure and this is an open structure. Now this dihydrothiamine can be converted with the help of water into beta uridoisobutyrate. Now what happens? So this can be done by the enzyme called as dihydropyrimidinase. So dihydropyrimidinase is an enzyme which can break the cyclic form of this dihydrothiamine into beta uridoisobutyrate. Now this beta uridoisobutyrate can be converted into beta aminoisobutyrate. So what's happening here? We are losing this ammonia. We are losing this ammonia in the form of NH4 plus ammonium ions. So this can be converted into urea from the urea cycle, and this is done by the action of an enzyme called as beta uridopropionase. Beta uridopropionase. See how many amino groups are there? 1, 2. There are 2 amino groups. Here we are left with 1 amino group. Yeah, this amino group has been removed. Now what happens? We are left with this compound. So this is called as beta aminoisobutyrate. Actually this whole part has been lost. This is lost as amino as well as bicarbonate ions. Now what happens? This compound, this beta aminoisobutyrate, can be converted into methyl malonyl semialdehyde. Methyl malonyl semialdehyde. You know this methyl malonyl semialdehyde can be converted to succinyl CoA, and this can be shunted through Krebs cycle for the synthesis of ATP. So this is how these pyrimidines are broken down. Here in this case we are going to use NADPH. These NADPH are synthesized from HMB shunt pathway. So in the pyrimidine breakdown or catabolism of pyrimidines, we are going to use this NADPH. Next, So if there are any genetic aberrations in the human purine metabolism have been found, if there is any genetic aberrations, then there will be serious consequences. For example, this adenosine deaminase or ADA enzyme, if this is lost, it leads to a severe immunodeficiency disease, which is called as a T lymphocytes and B lymphocytes do not develop properly. So if there is any lack of this enzyme, which is called as adenosine deaminase, if there is no adenosine deaminase or any aberrations to this enzyme, then we don't form this T lymphocytes as well as this B lymphocytes, and this lack of ADA leads to a 100-fold increase in the cellular concentration of DATP. So this is a strong inhibitor of ribonucleotide reductase. So if this is inhibited, then high levels of DATP produce a general deficiency of other nucleotide triphosphates in the T lymphocytes. If there is no T lymphocytes, then we know that these T lymphocytes and B lymphocytes, they are responsible for the memory, because if they don't have the memory, then they cannot synthesize the antibodies. Because these T and B lymphocytes, they are having memory, they are able to generate antibodies to the respective organisms. So if there is no nucleotide triphosphates, the T lymphocytes, then definitely we are going to lose their memory capabilities. So this will lead to an infection. A lot of infections will occur. So individuals with the ADA deficiency lack in an effective immune system, and they do not survive unless isolated in the sterile bubble environment. So every time they have to be in a space suit, in a special type of suit, such that there will be no attacking of any organisms, and this is a very high risk. In more recent trials, some ADA deficient individuals registered normal immune function after being given a functional gene for ADA. So nowadays there is a technology which is called as a gene transfer or the gene therapy. So what they will do, they will do the replications in the RT-PCR, and then these replicated genes will be inserted, gene incorporation, through the gene therapy. Then they can synthesize this ADA and we can come out of this environment. The purines and pyrimidine bases are recycled by the salvage pathways. How they are recycled? The free purines and the pyrimidine bases are constantly released into the cells during the metabolic degradation of nucleotides. So we know that we are going to get, from GMP we will get guanine, from AMP we will get inosine. So these free purines are in a larger part, they are salvaged. So we know that there is a salvage pathway where this will use the intermediates of the catabolism process. So they are salvaged and they are reused to make the nucleotides in a pathway which is simpler than the de novo synthesis. Because de novo synthesis takes about 11 steps to synthesize, taking the skeleton from each and every atom, like the aspartate or formates, folates, glycine, aspartate. So a lot of things are involved in the construction of the skeleton of the purine nucleotides, but in the salvage pathway it is not required. So all these 11 steps are bypassed. So just by from the catabolism we are going to get guanine. From that guanine we are going to get, with the help of an RPPP, we are going to get AMP or PI. If it is a guanine plus RPPP. So RPPP means phosphoribosal pyrophosphate. So this will combine with the guanine or adenine to bring AMP. So it's a very simple process. One of the primary salvage pathways consists of a single reaction catalyzed by adenosine pyrophosphoribosal transferase. So what we will do with adenine and this PRPP, we are going to get AMP. It's a single step. So this is what is called as salvage pathway. So as you can see here, it's adenine pyrophosphoribosal transferase. So this enzyme will transfer pyrophosphoribosal group upon to this adenine group. Thereby we are going to get AMP. So this is a 5-phosphate and ribose. So it is directly attaching this group to this adenine. So this is the action of this enzyme. Now what happens? In the same way we can synthesize guanine. But if we want to synthesize guanine, we require hypoxanthine guanine phosphoribosal transferase. This is what is called as HGPRT. HGPRT. So this HGPRT is very much important enzyme because through the salvage pathway we can recycle this guanine. If we have to recycle this guanine or hypoxanthine, definitely we have to synthesize, we should have an enzyme which is called as HGPRT. So likewise I told you yesterday, if we want to synthesize a GMP or IMP from guanine or hypoxanthine, definitely we require this HGPRT. So, and one more thing, in case of the synthesis of monoclonal antibodies, if we want to synthesize monoclonal antibodies, then we have to suppress the remaining kind of antibodies. So if you want to suppress the remaining kind of antibodies, then what we will do, we will grow in the HAT medium and HGPRT medium. So if we grow in the HAT medium or the HGPRT medium, definitely we are going to eliminate the unnecessary growing of remaining cells. Only this particular type of cells will grow and they will give out only monoclonal antibodies. So this is a principle in the creation of monoclonal antibodies, where we are going to grow the cancerous cells, because if we want more number of cells, then definitely we have to induce cancer, and from that we are going to induce these monoclonal antibodies. So you can study this extensively in case of the technology, biotechnology. So if you study this pharmaceutical biotechnology, in that subject you can study it in an extensive fashion. So but for now, if you want to recycle this guanine, we require HGPRT, and guanine can be recycled as GMP. In the same way, this hypoxanthine, because we see this adenine, guanine and hypoxanthine in the catabolism process of this GMP or AMP. So this hypoxanthine can definitely lead to xanthine. From xanthine we are going to get uric acid. So this salvage pathway can be creative, or it can be extensively use the metabolic intermediates like adenine, guanine and hypoxanthine. So through these intermediates, this will directly create AMP, GMP or IMP. So the salvage pathway of purine nucleotide synthesis with the help of RPPP, ribosyl pyrophosphate, or for example, so RPPP is involved in all the cases where adenine and RPPP are combined to give AMP, and guanine and RPPP are combined to give GMP. Hypoxanthine and RPPP can be combined to give IMP. The deficiency of HGPRT will causes Leishman syndrome. Leishman syndrome, because the salvage pathway cannot recycle this guanine or hypoxanthine. So this is what is called as the Leishman syndrome. Next, so this free guanine and hypoxanthine, the determination product of adenine are salvaged in the same way by the hypoxanthine guanine phosphoribosyl transferase. So a similar pathway also exists for pyrimidine bases in the microorganisms and possibly in some mammals. So a genetic lack of this HGPRT activity is seen almost exclusively in male children, results in a bizarre set of symptoms which is called as the Leishman syndrome. So if there is a lack of this male, in case of male children, if there is a lack of this HGPRT enzyme, then definitely it leads to a set of symptoms which are called as the Leishman syndrome. So there are certain devastating effects with the help of this, if we are lacking of this enzyme, that is the Leishman syndrome, then the importance of the salvage pathways will be lost, and this hypoxanthine and guanine rise constantly from the breakdown of nucleic acids, and if there is an absence of this enzyme, then even this PRPP levels will increase, because the salvage pathway will club these two to make new nucleotides, and these purines are overproduced by this de novo pathway, which is resulting in the high levels of uric acid and the production of gout-like damage to the tissues. So gouty arthritis, we call it as gouty arthritis, accumulation of uric acid in the tissues. So the brain is especially dependent on the salvage pathways, and this may amount for the central nervous system damage in children with the Leishman syndrome. So this Leishman syndrome will lead to a damage of the central nervous system in case of children. So this syndrome is another potential target for the gene therapy. So Nowadays this gene therapy is becoming more and more a focal point of research. So this Leishman syndrome is also a target for the gene therapy, because through this gene therapy we are able to induce the ADA gene, which is constantly secreting this ADA and HGPRT. Like that we can induce a gene which is responsible for the synthesis of HGPRT or ADA, as we have seen in the previous cases. So this gene therapy is a growing research area nowadays. So this excess of uric acid, if there is presence of excess of uric acid, this may lead to the gout. So gout is a metabolic disorder which is the accumulation of uric acid in the joints. So if there is an elevated concentration of uric acid, and if this is constantly accumulating in case of this joints, then we get a metabolic condition called as gout. So in gout we have two kinds of gout, that is the primary gout and the secondary gout. So not only the joints, the kidneys are also affected, because there is an excess of uric acid which is deposited in the kidney tubules. So gout occurs primarily in the males. So predominantly in the males it is higher. Its precise cause is not known, but it is often involving an undersecretion of urate. So if you are unable to excrete urate, then it will lead to the creation of uric acid. So the genetic deficiency of one or more of these purine metabolism enzymes may also lead to a factor of some causes. So gout is effectively treated by the combination of nutritional and drug therapies. So the drug therapy what we are giving is allopurinol. So allopurinol is a drug which is given to treat this gout disease. So the foods especially which is rich in nucleotides and nucleic acids, such as the liver or the glandular products, which are withheld from the diet. So if you are a non-vegetarian, then you can experience it well. Some of those will specifically go for the liver, because those liver products are having high amounts of nucleic acids and nucleotides. So if we increase the nucleic acids and nucleotides, then we can so the primary gout. So it's a newborn error or metabolism due to the overproduction of the uric acid. So this is mostly related to the increased synthesis of purine nucleotides. So the following are the most important metabolic defects which are associated with the primary gout. So what happens is the glycogen or glucose is converted into glucose 6-phosphate and then to ribose 5-phosphate. So this is a HMB shunt pathway. Now there is an enzyme which is called as the glucose phosphatase. So glucose 6-phosphatase. So this will convert back this glucose 6-phosphate into glucose, and this can be converted into glucose 1-phosphate, or this can be stored as glycogen. Now what happens, if there is a deficiency of the glucose 6-phosphatase, it's an increased utilization of glucose 6-phosphate, which is a reason for the elevated levels of PRPP, that is phosphoribosal pyrophosphate. If we are lacking this enzyme, then it is leading to an elevated levels of ribose 5-phosphate. If there is an increased level of ribose 5-phosphate, then also it leads to gout. So these are all the metabolic disorders. Next, there is one more enzyme which is called as a glutathione reductase. So even if there is a glutathione reductase, or there is an increased concentration of glutathione reductase, then this will lead to the increased utilization of NADP+, then increased ribose 5-phosphate and increased the synthesis of PRPP. So these two cases will definitely lead to the primary gout, and this is especially in the newborns. Then you can see from ribose 5-phosphate we are going to get PRPP and glutamine, which will lead to phosphatidylphosphatidylamine amine, and thereby we are going to get hypoxanthine. From hypoxanthine we are going to get uric acid. Now if there is an increase of this enzyme PRPP synthase, if it is increased, so this is under the feedback control by purine nucleotides. So we can our body has a mechanism to control this enzyme, because as I have told you, feedback mechanism is the future products are coming back to inhibit the first step. Now this HGPRT, if there is a decreased concentration of HGPRT, which will lead to the enzyme of the salvage pathways, Leishman syndrome, and then it is an increase in synthesis of purine nucleotides. If there is an increase in the synthesis of purine nucleotides, then the degradation is also increased. If the degradation is increasing, then we got increased concentrations of uric acid. So this is what, and then there is one more step. You can see at this point, the PRPP glutamyl aminotransferase, even there is a lack of this feedback control of this enzyme by pyrinucleotides will increase the elevated synthesis of nucleotides. So what happens if ultimately, if the concentration of nucleotides is increasing, then automatically uric acid concentration also increases. If this accumulates in the joints, then we get a gout. So we should see that there should be the right level of concentrations of these enzymes. So now from hypoxanthine we can get uric acid. So allopurinol will inhibit two things. One, the xanthine oxidase, and this can halt two steps. One, the conversion of hypoxanthine to xanthine, and from xanthine to uric acid. So these two can be inhibited by allopurinol. So allopurinol will inhibit this xanthine oxidase enzyme, which can stop of hypoxanthine into uric acid. So if there is a less amount of uric acid, definitely this will not accumulate in the joint. Now we will talk about the HGPRT deficiency. So this is an enzyme of the purine salvage pathway, and this is the deficiency will cause the Leishman syndrome, as we have been discussing. So this disorder is associated with the increased synthesis of purine nucleotides by a twofold mechanism. So if there is no enzyme, this is the HGPRT, then definitely we are going to increase the concentration of purines and pyrimidines. So firstly, the decreased utilization of purines, there is a hypoxanthine and guanine by the salvage pathway, which will result in the accumulation of the diversion of PRPP of the purine nucleotides. So because this enzyme is especially available in the salvage pathway, then the salvage pathway cannot recycle the free purines. If the free purines are not recycled, definitely it is an increase of these purine nucleotides. So if there is an increase of purine nucleotides, definitely we are going to end up with a high concentration of uric acid. Now secondly, the defect in the salvage pathway will lead to the decreased levels of IMP and GMP, causing impairment of the tightly controlled feedback regulation of their production. So since the concentrations are decreased, then there will be no regulation. So these are the two effects of this HGPRT. Now again the same thing, this is related to the previous slide where I have shown a pictures of the various deficiencies and how they are going to affect the various metabolisms. Now what happens if there is an elevation of this glutathione reductase? So increased glutathione reductase generates more amount of NADPH. So this is utilized by the HMP shunt pathway. So this causes increased ribose 5-phosphate and increased concentration of PRPP. If there is an increased concentration of PRPP, definitely we are going to make more amount of nucleotides. If there is more amount of nucleotides, if they are undergoing degradation, then it increases the concentration of uric acid. So if this uric acid will deposit in the joints, then we get arthritis. So now the secondary gout. This secondary gout, which is also called as the hyperuricemia. So hyperuricemia is a metabolic disorder where there is an increased concentration of uric acid in the blood. So it is due to the various diseases which is occurring increased synthesis or decreased excretion of uric acid. So increased synthesis of uric acid or decreased excretion of uric acid. So these are the two main effects. Now increased degradation of nucleic acids, hence more uric acid formation. So this is observed in various cancers like leukemias and polycythemia and lymphomas and psoriasis and increased tissue breakdown. So because of the trauma or the starvation So if there is a necrosis, in such cases we are going to increase the concentration of uric acid formation. So these disorders are associated with the important or impairment of the renal functions, and these will cause the accumulation of uric acid synthesis, uric acid excretion. And if there is a lack of this excretion, then this will leak into the tissues, and then it will end up in the formation of a gout, a metabolic disorder of. Now, to alleviate the symptoms of this gouty arthritis, then we have to give allopurinol. So it is a substrate of xanthine oxidase which will convert this allopurinol into oxypurinol. So this allopurinol can be converted into oxopurinol. This oxopurinol inactivates the reduced form of the enzyme by remaining tightly bound to it. So this is inhibiting the enzyme. When xanthine oxidase is inhibited, the excreted products of purine metabolism are xanthine and hypoxanthine, which are more water soluble than the uric acid and less likely to form crystals. So if since the xanthine and hypoxanthine are more water soluble, they can be eliminated easily in case of urine.